Welcome to Hot Chips 29. Keynote 1. The direct human-machine interface and hints of a general artificial intelligence.
All right, guys, we're ready to start the afternoon session. Welcome back. With the eclipse out of the way and lunch out of the way, it's basically the fun is over and it's all just hard slogging for the rest of the afternoon, so brace yourselves. It's my great pleasure to introduce Dr. Phillip Alvelda as our first keynote speaker. Dr. Alvelda just finished a 3-year stint at DARPA, where he was program manager in the biology initiatives, running a program working on direct computer-human interface, brain interface. Very exciting stuff. I first met Phillip a couple of years ago. I was going to Washington, D.C. on other business, and I put up on my Facebook page, hey, I'll be in Washington, what should I do? And a friend of mine said, you got to meet Phillip. So I went to meet Phillip. And after he had shown me the Lego models of working engines, V8s and V12s and inline 4s that worked, we got down to looking at all sorts of amazing stuff of how you interface hardware to wetware. And it's pretty cool stuff. So I'm really looking forward to what he's going to tell us about that, and also some of the hints from that research and that work that he's done of where we might go next beyond what we understand today, which is so exciting from deep neural nets and that sort of thing. So please welcome Dr. Phillip Alvelda.
Thanks, Fred. Well, good afternoon, everyone. It's good to be back here in Silicon Valley. When we moved to Washington, D.C. with our 2 daughters for the DARPA gig, I was telling my wife, our older daughter's 14 years old. She's grown up in Berkeley. She's never met a Republican. And, wouldn't it be great if she could have an experience which broadens her perspective? Well, we didn't quite expect what we got. Our experience now is very broad. We're thinking about perhaps fleeing to a bluer state now, but I'd say that the DARPA experience was fantastic all around. And what I hope to do today is give you a little bit of a flavor of how a DARPA-type technology initiative can come about, what it's like to be on the inside of it. And I think for the Hot Chips community, what I'd like to offer is a little bit of a different talk. Rather than talking about specific technologies and specifications, what I'd like to do is give you a view that's looking forward a little bit more in the future for what are the types of technology, data rates, data formats, information theory, application challenges that we think could really drive new capabilities in machine learning and apply some of the things that we learned in trying to get artificial electronics, in a way, making artificial intelligences in order to mate them to our real intelligences. And how we can take some of those learnings and apply them to create more capable, more anthropomorphic systems in the future. So my talk today really is to introduce this notion of new combinations of carbon and silicon. Coming out of DARPA, I've started Cortical.ai really to take the technologies and the approaches of what are the new types of computation, what are the new types of systems, what are the technologies that we required to make those interfaces possible and actually function, and apply them to building artificial people. And in the company, we really focused on a couple of different areas. Most notably, the thing that I'm going to concentrate on today is the inside. What is the computing element, the thing that binds us together cognitively, that allows us to do very, very powerful things, some of which are still out of reach of traditional processing systems as we manufacture them in electronics and photonics. We also work on what is the outside and how you can apply more anthropomorphic computing systems to make more realistic animated figures and renderings. But you guys are going to get a lot of that from the video game and graphics folks, so I'm going to leave that out of this presentation and concentrate on the guts of the technology. So in the sense of neural technology, what is that machinery inside the head that makes us so powerful computationally? Now, the journey for DARPA started long before I got there, back in the 2002 era, where we began investing in prosthetics for wounded warfighters, people who had lost a limb. Obviously, for a couple hundred years, we've had the hook for the arm, the peg for the leg. But the investments in Dean Kamen's DECA research drove what we call the Luke arm, in homage to Luke Skywalker and with his prosthetic at the end of the third movie. But here, this was an 8-pound prosthetic with a modular system. You could have, elbow, wrist, shoulder versions. And when it operates, it's really quite remarkable. You can see that these folks who have lost, different types of appendages, depending on their wounds or congenital defects, can actually control quite accurately what they do with the system. They can pass grapes back and forth. They can take these flimsy water bottles and drink from them without squirting the water out. And such an incredible leap beyond what's happening. I'm happy to report that now with the Veterans Administration, these things have an insurance reimbursement code and are being distributed, and the volume is growing year on year to what we distribute to our wounded veterans. But in a sense, this was a fantastic first step, but as in many of these demonstration videos, one of the key issues, of course, is what's not on the screen. And what you don't see is the system that's actually controlling the function of the robot arm, which is a mechanism that straps to the shoe. And it's got a gyroscope and some buttons built into it. And so all of the sophisticated functions that you see happen there is mostly the derivative of clever programming and foot motions and pedals while the person is learning to use the prosthetic. So that really isn't the Holy Grail. The Holy Grail, of course, is to take something like this and connect it to the peripheral or central nervous system so that, like Luke Skywalker did, you could control it with your thoughts. Now, that journey at DARPA began, of course, with animals, and this is out of Miguel Nicolelis' lab at Duke University, where hidden behind the aluminum block there, you can see that the monkey has a box with some wires coming out of it. There is a probe of wires that is really shoved directly into the motor cortex of the mouse. So the area of the mouse's brain that ordinarily sends the signals out from the brain to control its own arms. But you might notice that its arm is currently trapped in that transparent sleeve there, and he's being presented the marshmallows, and simply with his thoughts, he is controlling the robot arm to grasp the marshmallow and feed himself. So he's using his visual system, targeting his grasp, maneuvering the fingers and the arm to grab the marshmallow and give it to himself. Now, couple profound realizations. This involved, again, what's not on the screen, the giant refrigerator of computing, the big bundle of cables, the screw plug in, to transit the skull. Quite a lot of effort to digitize it, very, very high rates with 12-bit DACs, getting those nervous signals, very noisy. We'll see a little bit more, but this was the first time, this was the kind of the watershed moment where a creature controlled an electronic system with reasonable degrees of freedom, call it maybe 2 or 3 degrees of freedom to control this particular arm, completely under mental control. It wasn't long before that eventually came through and was used in humans. Some of you may have seen this 60 Minutes video.
And straight down.
Jan Sherman, quadriplegic, paralyzed from the neck down.
Left and right and diagonally.
You can see the box on her head.
I can close it and open it, and I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you come over there.
Okay.
You grasp your hand there. There we go.
Oh, my goodness. Wow.
And I can do a fist bump if you'd like.
That's amazing.
And it really is. It's amazing. So that was the first moment that a human was able to control an electronic system on its head, just with thought. Again, what's not in the screen? The giant refrigerator of computers, the big bundle of wires, the refrigeration system for the computers. And the fact that within the system control the arm, it had quite sophisticated digit control, but those were all manufactured from watching people and how they grasp things habitually. There's really only about 5 or 6 degrees of freedom in that robot arm, and it was still slow. It's not something that you could have individual control of all the joints in the fingers as you might want if you were to try and play a piano, and it certainly didn't operate fast enough to catch something that was thrown at you that we do quite easily. Since that program, one of the first programs I had the opportunity to work in coming to DARPA was the haptics program that was generally run by Doug Weber. I got to assist him on several of the different projects. But here, the idea was to accomplish the return path. So instead of just controlling the robot arm with your thoughts, the challenge for Jan Sherman and the other operators was to use that system. She had to concentrate very heavily and look at the arm because there was no built-in feedback like you have in your own body. There's no system where, without looking, you could tell where your joints were or where your limb might be. There was no feedback when you bumped into something or touched something. How hard were you grasping? So this was the project to build the electronic sensors into the digits of the robot, to build the proprioceptive sensors in the joints to feedback the position. And I'm happy to report that this has been a fantastic success, where you can see some of the early tests here of a wiring directly into the peripheral nerves in the root nerves of the arm that remain after an amputation. And there are similar systems now which we have accomplished the same functionality directly into the cortex. So both to the peripheral nervous system and to the central nervous system. But the really remarkable thing about all those experiments is that as wondrous as they were, as much of a watershed moment as we saw in that first opening of the door of controlling things with your thoughts, the state-of-the-art technology, now keep in mind, those early demonstrations with Jan Sherman, 2005. Last year's haptics demonstrations with the return path and the sensor, early 2017. In that entire span, this remained the state-of-the-art technology that the FDA would allow us to implant in a person. Some of our technical staff at DARPA would laughingly joke about it and say, oh my God, stone knives and bear skins. It's nails and nerves. We're taking this thing and, shoving it. I mean, of course they do it carefully and they've got machinery and they clean it and all that. But the point is, they're just pushing this little pad of 100 wires, a couple millimeters on a side, and getting a really noisy signal out of those 100 wires. And out of that, we eke out on the order of 6 degrees of freedom, and we can kind of slowly and herky-jerkily control a robot arm. So I come to this situation and say, my God, the technology hasn't advanced really since 2005, and really that's probably like 1980s level technology, right? What could we do if since 1958 we've been trying to hook up electrodes to the brain? This was an early experiment over the visual cortex where it's an array of rather large, couple millimeter sized electrodes. You can see the pattern on the right where they were drawing in the notebook the dots that they would make in their vision. This idea of phosphenes. You put an electric current right over your visual cortex, you get this bloom of light. We've known since that time that electric stimulation was not ideal. Yes, you'd get a visual response, but you weren't speaking the language of neurons. You were just pumping a bunch of current in, you're kind of setting them all off together. But that agglomeration of neurons, each one of them has a different computational function. They represent different things, even though they might be right next to each other. Some should stimulate activity, some would inhibit activity, some are different colors, some are orientations, some are motions. And when you just plug the current in and turn it on, yes, you get activity, but it's not in the language. It doesn't have the precision to represent how finely we know our eyes work. So, of course, I come to DARPA with my background as a physicist and an entrepreneur and an engineer, and I look at this system. You've got trillions of synapses on one side, trillions of transistors and wires on the other, and so far we have, 100 wires? That's a nice 10 order of magnitude bottleneck and ripe for exploitation. So I see the opportunity to apply decades worth of technologies to think about how would you develop a next generation of neural system interface specifically for use in humans with a rather audacious goal. I wanted a 4 order of magnitude improvement in the scale at which we could have that interface. Now, when we first came up with that number, we actually did it with a pretty rigorous study of, bottom up. What is the fundamental transductional element in the neuron? What is the amount of energy required to change its state? What would we need to transduce it? What would we need to process it and encode it? From a first principles perspective, what did we think was possible? 4 orders of magnitude is a big jump. There's a huge motivator to do it. And this graph was one we did to get the project approved, where we're looking at the spatial precision on the bottom axis, the number of independent channels of neural axis vertically on a log scale. And you can kind of see that we started the DARPA project with the types of technologies of animal experiments where we could demonstrate very, very limited scale, very limited independent channel count. And there were some things where we had larger scale. You could use MRI to see many neurons, but you couldn't see many independent channels. You would have very, very large voxels. But of course, the goal, is to have more precision and have more scale so you can do more complicated things through your interface. You want higher bandwidth and higher precision at the same time. That was the core requirement. So the question is, what was a reasonable target? Well, there are some new technologies that would take what had been done in humans, that's in the diamond boxes, apply some of the latest technologies that had been demonstrated in animals with more scalable electrode systems, but they hadn't been pushed through the FDA. And there were some exciting new technologies which came out, which were the catalyst for me to come to DARPA. It was in reading the Ahrens and Keller Nature paper on how you can make not just electronic systems, where, to get to the auditory cortex, we could just do a DARPA hard program, apply the very latest kind of 2015, 2017 era technology nodes in electronics, and have something that could achieve what we thought was about the scale of the auditory cortex. But the new technology catalyst, the idea of optical interface, was super interesting. And we believed that those early experiments on fish and rodents and monkeys would offer opportunities at tremendously larger scale. And so this was really kind of the product sell sheet for the DARPA management. It is, we think we can move the state-of-the-art human interface from the coarse motor control era to the fine sensory interface scale. So 3 years back, this is what the technology looked like. This is out of Mark Schnitzer's lab right here in Stanford, where he was looking at a screen. Now that vertical silver bar, that's an aluminum track, and down at the bottom you can see there's a tiny mouse. And it's got an ultraviolet microscope, imaging light and fluorescent microscope on its head that's looking at the part of its brain that has the place and grid cells. So what you're seeing here is one of the first demonstrations where we're starting to decompose what is the spatial and temporal and physical function computationally of the neural substrate. And here you can see the tiny little circle there that's circling a neuron that corresponds to the triangular point towards the top of the track. That neuron fires when the mouse passes that triangular point in a specific direction. So it's direction and position sensitive. And so you can see the mouse run, and you can see the total energy inside the circle in the bottom graph, and you can see every time it passes that little triangle, you see the blip of activation. And that whole area of neurons, that is encoding every position on the track. And by hooking up a machine learning system, they could actually turn off the camera, just look at the neural representation inside the mouse's brain, and tell where the mouse thought it was. How cool is that? This is some work out of University College London, where this is also in a rodent brain. They are doing a bidirectional read and write. And so now these technologies of making the neurons optically active, this is work that was derived from the ability to program with DNA. So synthetic biology, crafting an instruction that you administer to a cell, it creates a protein that when the neuron fires, calcium rushes in and it will fluoresce. So that's how you read. You write by having a mirror, an analog to that, where when you shine light on the cell, it opens up a membrane channel, forces ions to flow in, and the neuron will fire. So we have an optical interface system, which they can now use. In this case, it's a scanning system, where they will pulse the laser and add energy in a linear sequence to all of the circles in that area of the rodent brain. And you can see that even stimulating some subset of the neurons in the area through their dense interconnection to the neighboring neurons, you can see other neurons light up. So in a way, this was one of the first experiments to, in this case, the area of the mouse brain that controls the whisker sensation, where just with light, they could write a code that would give an artificial sensation to the mouse. 3 years ago. This is last year. Also Mark Schnitzer's lab. So significant progress. We're looking at the entire cortex of the rodent. Think about that. We're looking and seeing the individual neurons computing as the mouse is awake and running on a treadmill. You can zoom in a little bit to see some of the individual neural activity. And here it is with a little bit of image processing, where you have no difficulty making out the individual neurons. So one of the obvious applications for machine learning in this field was, we don't understand what all of those flashing lights mean, but we can watch the behavior, we can instrument the mouse, we can look at what's happening computationally and develop correlations and start to tease apart what is the actual neural code, what do these things represent, like the position of the mouse in the track, but now we're looking at putting images in front of the mouse and reading out how the brain is processing those images. Now, when you start to look at neurons in this scale, and here we're just starting to zoom in on one particular bit of the mouse brain, you can kind of see some of the areas of the brain that are illuminated according to the Allen Brain Atlas for the rodents. But you can start to see what the machine learning systems will do, where it will isolate and identify and tag each individual neuron and its isolated activity. And you start to transduce what the energy is in each one of those circles, and you begin to develop a representation for what is the neural code, when are the neurons firing, what are they related to behaviorally. But think about that. We're going from about 100 wires to being able to transduce, what, in this case, 1.5 million neurons? That imposes some challenge. Now, the brain system that we hooked up to Jan Sherman was already kind of at the limits of scale that we could manage. And so state of the art was to take that 100-wire implant in the motor cortex, and think of it, these wires are very, very coarse with respect to neurons. Any one of those wires is surrounded by some between 20 and 50,000 neurons, all actively going and generating electrical signals. So you end up with something that is not really an identifiable code of any sort. It's an agglomerated signal with a tremendous amount of noise. So computationally, this was very, very difficult. We would sample it, at 30 kilohertz. We'd use 16-bit D to A's over 96 channels. So that's a 60 megabit data stream just to extract 6 degrees of freedom. And the code that would drive the robot arm was only driving about 5 kilobits per second of data to control the arm. The arm had a lot of smarts to do the finger control and grasp and other things, but this was the kind of sparsity of what we could extract from that very, very noisy signal. But now the challenge is when you want a sensory interface, we're talking about going to see what does the architecture or the system look like that would be able to transduce and transmit millions of channels. Well, the beauty is, if you were to try and do the same thing that you were doing with the larger scale system, it would be intractable. But if you think about making a microsystem, where the scale of the transduction and the accuracy of the interface is closer to that of the neurons themselves that you're trying to speak with, now you've got something where you can begin to isolate the individual activity of individual neurons, which is very, very compressible. So we realized at one point that just to say we want 1 million neuron interconnect was only half the challenge. You needed, in order to be able to make the problem tractable, you had to simultaneously have the precision to be able to make a compressible signal in a manageable data stream. But there are bigger questions. Even if we can start to do dimensional reduction and say we're just going to track with high precision when a neuron fires, it's an impulse function, and we can make a code that looks like this, where you translate from an image to 100, in that 100 sampling unit interval into streams of pulses. We actually need to develop these transcoding algorithms which could efficiently go from what is the neural representation to the representation of the sensory media that is causing that stimulus. And of course, our goal was to make an artificial system where you could take any stimulus, whether it's a real camera feed or a synthesized image, and be able to translate it into this pulse neuron code and write it into the brain so you could create a real interface. But then you start to say, well, what is that code, and how do you do the decomposition? And the more we started to look at the fundamental principles of how you represent information and how the brain was doing this computation, we realized that our first guesses, and really what neuroscience has been using for the last 60 or so years to represent neural activity, wasn't telling the whole story. And so for you, double E folks, Shannon-Hartley digital channel capacity, this is a very familiar graph where, for example, you take a bundle of neurons, say your optic nerve, and you count them up. There's about 2 million of them. And then you say, well, how fast can a neuron fire? That's kind of my switching rate. You look at what the magnitude of the signal is, and you figure out what the background noise is, and you have a limiting channel capacity. Makes sense. We did those calculations. But then how do you apply it to a neuron? In the neuron, you've got kind of the neuron that's going to fire a signal. It fires it down the axon to the right where those blue junctions are to the other neurons on the other side of the way. Your temptation is to say, well, let's just use Shannon-Hartley. The axon is the communication channel. There might be some background noise. You've got the impulse of the nerve. We calculate what the channel capacity is. We count firing rates in the nerves. And we end up with a channel capacity description of about 2 bits per neuron-ish. Well, turns out that's completely wrong. Or wrong is probably not the right way of saying it. It's an inappropriate model. And we have the proof. Because we can do behavioral experiments and calculate what is the information necessary for you to discriminate patterns that are very close to each other. And the two most obvious ones are discriminating 2 tones in your auditory system that are very, very close. And here you can kind of look at the lines of what Shannon-Hartley says you should be able to detect, which is the blue line, and the finer resolutions that people seem to be able to detect, which shows that you can do a couple orders of magnitude better with your biological system than Shannon-Hartley would seem to allow. So if we were to describe the code the way Shannon-Hartley describes a digital system, it's clear that that's not capturing the information that's in the channel. Same is true with your visual system. You look at whether you can discriminate 2 lines that are close to each other, same calculation, same result. You're about 2 orders of magnitude shy compared to what your body seems to be able to do. So where's the missing information hiding? Or a different way of saying it, what aspect of the coding or the transmission channel or the computing that's happening in the channel are we not appreciating in our description of the thing as a simple digital channel? And there's a few general areas where we believe there's a good amount of information that could be derived, but it turns out that the one which seemed to hold the most was in the code being not a code of signal level and switching, but in time of arrival. And in fact, not even time of arrival of a single symbol, but the relative time of arrival of multiple signals in a shared channel. So these distributed codes, these population codes that are time of arrival based. And the experiments that back up this idea told us some really interesting things. This is a beautiful, simple experiment by Incy, who's now at Edinburgh, where he took electrodes and put them on both sides of an axon channel, sampling many of them in parallel, and he began to do information content measurements and entropy measurements to determine what was the information in the channel relative to the sample rate. Now, for 60 years, most of the neuroscientists would tell you, 100 milliseconds, 20 milliseconds, 10 milliseconds, that's a fine sampling rate for our patch clamp experiments, seeing what a single neuron does. But look at this. As you decrease the, let me go back. As you decrease the sample rate, the information rate goes up, and the experiment broke when the system failed at 2 kilohertz. And we're kind of at an increasing slope of deriving more and more information. What this told us was that if all you look at is a spectral decomposition or an average firing rate, you're not getting the majority of the information in the neural signal. So now we find ourselves at kind of the uncomfortable junction of high spatial requirements, high temporal sampling requirements, and a desire to take these big boxes that are bolted to the skull with large bundles of cables and saying, I want to integrate this in a package that you could insert in a human. So what's the solution? We realized we're screwed, totally screwed. We're never going to get to 1 million neurons. We'll make something that samples at 30 kilohertz. It's got megapixel resolution. We'll put it on the cortex, temperature rise of maybe 30 degrees. FDA will allow 1 degree of temperature rise inside your head. So boil the cortex, probably a negative outcome. But we realized at some point that obviously the brain was doing it. We know that your brain, even actively thinking very, very hard, eyes working, ears working, all of your cognitive processes going, peaks at about 25 or 30 watts. It's a remarkably efficient machine that's computing at gigaflop rates for 30 watts. So for the area that we want, say we wanted to put a device over your visual cortex, surely we could make something in the power budget if we but designed it to operate more in the way that the human brain works. And so here the key situation was to realize that the brain was not a synchronously clocked architecture. It used a spatiotemporal code, but it was sparse and it was asynchronous. And those 2 features, it turned out, was the key. And so we began looking for technologies that would allow us to incorporate sparse asynchronous coding in both the sensors and the computers to build these integrated devices that would receive the possibility of getting to the scale we wanted. And so the first one we found, there's a few examples that DARPA had funded in previous programs. This is the ChronoCam group out of Paris, where they're looking at kind of the traditional limits of a synchronous clocked architecture, where for power reasons, you just can't have simultaneous scale and sample rate. And so they went to a different architecture which had a much more complicated pixel that would detect changes and sample only when there was a change. And then, of course, they were able to preserve microsecond scale temporal resolution at megapixel densities at very, very, very low power. So knocking 2-3 orders of magnitude off the power budgets specifically for sparse images. And so this architectural transition, it's one of those great technology situations where you realize that your limitation was only because of the architectural assumption you were making for historical purposes. And if you could just change your perspective a little and go to an architecture more like the brain, boom, new avenues. And the applications are pretty cool. You can kind of see these artificial retina type of things that you may have seen here and about. That's Christoph Posch, one of the designers of the camera. The applications range for things where telemetry and power are limitations. You can kind of see the traditional camera in the top left, and then the 2 channels of the asynchronous temporal code camera in the bottom left is the event. I think the top right is the events, and the bottom left is the illumination measurements that are made upon those events. And there are clearly applications where this would start to fail. So, it's the traditional MPEG encoding problem. If you're watching the NBA basketball game and the HD camera pans across the screen and everything is changing at once, the visual system would be overwhelmed. The buffer of events would stack up and you'd lose imagery. But guess what? That's exactly what your eye does. You can't pan across the basketball screen any better than your TV. You're fooled when you're watching the TV because you've got a fixed gaze and the TV is moving. So you kind of see that particular edge case. But this would fail in exactly the same way that your eyes fail, which make it a great interface technology for direct access to the brain. There are other applications where sampling precision is very important. And when we started to look at the application of how do we extract the most information from the neural code, this was specifically the feature that we were looking for. We wanted very, very high temporal precision with very, very low power. And you can kind of see the asynchronous camera on the left, the conventional camera on the right, and the power consumption is about 1/1000th on the asynchronous camera, while you're getting better tracking precision in the imagery. It also is designed so it has very, very high dynamic range and functions in a couple of other different modalities that make it useful for other military applications like target tracking and things of that nature. But for us, it was the key technology in how we could extend the brain interface to scales that were meaningful. So from a company spin-out perspective, I decided upon leaving DARPA to start Cortical, specifically to apply some of the things that we learned in trying to figure out how do you interface these systems to natural biology and have meaningful information exchange. The first core idea, the first opportunity, is this notion of how can you use these temporal codes to build richer interfaces at much lower data and power rates. So we see lots of opportunity there. But beyond that, you start to think of packaging. You know, when you say, I want a general neural interface, how do you avoid boiling the cortex so that wherever I might want an interface to any of the different sensory systems, I want to be able to put a package that's wireless, that can be fully implanted, no wires transiting the skull. That turned out to be the limit of all our experiments, the open wound where the wires were coming out. We had to take them out for those people after about 6 months of experimentation. But the packaging is evolving incredibly rapidly. From the first experiment a few years ago of the light sheet microscope on the optical bench, the first kind of modular commercial product with kind of the kit optical parts, and then the hand-machined graduate student project for the smallest, at that time, fluorescent microscope that now is a spin-off called Inscopics in the Palo Alto area, from Mark Schnitzer's lab, where there's probably now half a dozen Nobel Prize-winning labs that are using that technology to understand what the brain is doing. And of course, DARPA looks at that and you say, well, that's a great graduate student product, but the FDA is not going to approve inserting that into human brains. But all of those technologies, the webcam, the optics with the filters, the illuminator, all of these things are systems which we can replace with planar photonics now. And so we believed it would be possible to take these types of systems and make a program where you have effectively a universal neural interface, which is a modular device, fully self-contained, wireless, and can perform the same function for the brain that the modem performed for the early days of the computer and the development of the ARPANET. It standardizes the interface to a system that otherwise is inaccessible. The goal, of course, being that we want to access whichever part of our brain we'd like to connect to at very, very high data rates with high information capacity. So part of the fun, of course, of being at DARPA is that everyone takes your call because you've got a lot of money. And I took this opportunity to realize and discover, much to my frustration, that all of the technologies that were necessary to make this sort of leap in interface were possible, but they were stovepiped and they were held by all kinds of different companies, and generally university researchers did not have access to them. And even when the universities had access to them, they would be working in their own discipline, but to make all of this kind of an integrated system operate, you needed, computational neuroscience, photonics, electronics, microelectronics, regulatory, packaging, computer science, electrical engineering, fabrication, the whole 9 yards. It was a huge reach just to get people to talk to each other and realize they could manage their part, but if they would but circulate amongst the experts, they could assemble systems. So really, the challenge more than anything was ecology and ecosystem building, where we needed to tell people if they but could get together, great things could happen. So we hosted over the course of 3 years a series of workshops, and ultimately we defined the program with a huge pot of money, and we set out a very, very challenging goal of this next generation interface. And it turned into a kind of a 3-continent effort. And these were the entities that we funded. We probably engaged about 10 times this number and visited over 80 labs to learn who could do what of the systems. And so let me tell you a little bit about some of the systems we funded. Now, these are still works in progress, so this is a little bit of a sparse report, just to tell you generically what are the technologies that are being applied to address the problem. But here, this is a prosthetic that's being developed by a team led by Ken Shepard at Columbia University, where they're making thin CMOS arrays that are purely electronic. So they're so thin, in fact, they're almost tissue consistency, but they have a completely integrated system in that one CMOS device. It includes power reception, telemetry, bidirectional telemetry. It involves the neural sampling grids, neural stimulation grids, and would take the types of technologies that we see in kind of epilepsy localization, where they've got the big patches that they put over large parts of your brain, and take it to the kind of individual neuron resolution. So this is a fascinating project. It's very, very integrated, but you can just see the participants in the program span universities, startups, large medical and regulatory agencies. Here's another project by FVE in Paris, the Foundation of Vision and Hearing, in collaboration with Stanford, the Friedrich Miescher Institute in Basel, the Chronocam company with the asynchronous sensor, GenSight, which has a technology where they can deliver genetic instructions to make individual types of neurons, cell-specific instructions, active optically. So here you can see on the right these illuminated, colored parts of the retina. They were able to send different genetic instructions to the different retinal components and activate them independently. And so the idea here is cortical sight, or a LED stimulator for an optically activated patch of the visual cortex. Fascinating. Here is a similar project run by the J.B. Pierce Laboratory, Vince Peerbone at Yale University, in collaboration with Jacob Robinson and Caleb Kemery at Rice and a group at St. Andrews working on the LEDs. This also is using a thin CMOS device, single integrated, with 3D epi liftoff 3.5 materials for the illuminators. So the idea is to be able to read optically and write with the LED focus. But one of the real innovations here is not just to light up an area and have the neurons fluoresce, but to actually write genetic instructions to have the neurons generate their own light, like fireflies. And in this case, they're using DNA from ctenophores, these tiny little kind of mini jellyfish that generate their own light. So the idea here is we use synthetic DNA programming to write the instructions that whenever a neuron fires, it generates light. This actually solves a bunch of problems, but it doesn't generate very much light, so you need a very high sensitivity detector. This is where Columbia comes in, where they are developing avalanche photodiode detectors that would be integrated in CMOS. But again, single monolithic device with a relay patch external to the head. This is a graph from the St. Andrews folks that are generating the high-efficiency blue LEDs. This is the wavelength that's very well matched to the sensitivity of the opsins that open the ion channels in the neurons. But you can kind of see the level of integration and the different technologies that are necessary beyond understanding the coding, which is you need the integrated photonics, you need the integrated power management, the integrated compute, integrated memory, all of these, and of course they need to be packaged and go through the regulatory machinery. The detector, the optical detector, is also very interesting, and this is work out of Jacob Robinson's lab at Rice, where here the challenge is you don't have much offset from the neuron to the detector. It needs to be flat because you don't want an unsightly bulge in the skull or an open wound of any sort, so it has to be a flat system. But you also want precision focus and high numerical aperture to collect all the photons that are coming off of those fluorescent materials. And so they developed this phase mask, which can essentially make a 3D sensor. It's fascinating. Here's a video of it operating on a 3D surface. It's a little bit of a confusing view graph. The bottom right green thing is the actual pattern that's being sensed directly by the camera. And you can see the camera positioned at the top looking at two different channels in an agar target to simulate the human brain, and they pass fluorescent beads through the two channels at different depths. And the point is that you can see the reconstructed images from the computed functions of the imagery, where you're combining the computational power of the sensor and replacing the optical element so that there's no relief or size in the box of the optics that does the sensing. So you have a new type of optical microscope which can lay flat like a tissue on whatever it is that you're sensing and have very, very high precision microscopic images. That's what we need to look at the brains. A similar project optically is also being conducted by Berkeley and the University of Paris and Argonne labs, where this is more of a standard architecture with a spatial light modulator and a camera in a folded configuration. This one, is probably more suitable for experiments. It's got an open skull with a screwable filament and aperture. But there's still going to be some technical challenges for passing that through the FDA or making new systems that are small enough to be implanted. Now, one of the other challenges is that up to this point, the things that we talked about funding were monolithic units. So for parts of the brain, like the visual cortex or the auditory cortex, where you want very, very high precision over a very well-defined area, the monolithic units seemed appropriate. But there are all kinds of therapies and interfaces that we could imagine building that need a much more distributed contact with larger areas of the brain, where we don't necessarily have a good idea for how dense the connection needs to be, because we don't understand what the coding is in those parts of the brain. And we know that, for example, in speech, the centers that govern speech, where if we want to look and have an interface to essentially read what you're imagining saying from your brain. So imagine you have a relative or you yourself are one of these locked-in people that are in a vegetative state, but they're actually conscious underneath. They just can't control their musculature to communicate. This is a sort of system where, with a distributed set of contacts, we could be able to parse what they're imagining saying electronically. And this system from Paradromics, it's a spin-out of Krishna Shenoy and Nick Mellish's lab at Stanford, collaboration with their folks there, the Francis Crick Institute, Cal Berkeley, Bob Knight's lab there, Kai Lesti doing some circuit design, Cirtec doing some regulatory and contract medical device manufacture. You see a system where they can take a series of optical or electronic fibers with glass cladding, bundle them, polish the interface, and abut it to a CMOS sensor or to an array to write. And then you've got these flexible fibers which you can then distribute and that are flexible enough to not just penetrate and damage nerves and blood, but be able to kind of wiggle their way in and contact the systems. But this is fundamentally an electronic interface with a long wire to the neuron. The last one that's also very interesting, led by Brown University in collaboration, this is by Artur Normico, with Stanford, the Wyss Institute in Switzerland, the Salk Institute in San Diego, and Qualcomm for the wireless systems. This is almost like a mini cellular network for a shower of tiny miniature neurograins, which are distributed and scattered across the brain interface of interest with a relay patch on the top of the skull. So this is miniaturized systems that have power delivery, backscatter-based low-power telemetry, and all of the neural sensing and stimulation technology isolated in individual grains. Now, what was really interesting about this one was, first of all, being able to scatter it over wide parts of the cortex and interface with larger parts of the brain. But here, you think of brain-machine interface as being an application where, you can read stuff out and then write stuff in. But here, you could have an immediate jump relay where if, for example, you've got schizophrenia, which we now can look at the physical underpinnings of the psychiatric disease and say there's a deficit in connectivity between two brain regions, we can make artificial neural links to connect disparate parts of the brain that have a missing component. So there's a whole series of things that we think we can both correct and possibly even enhance, improve the connectivity between parts of your cognitive processes. Kind of a tantalizing thought. So the great news, though, and really one of my stated goals coming into DARPA was that by creating this ecology and spending the last 3 years kind of getting the right people together, getting the right industries aware that they have a role to play and opportunities in this field, even though it might seem quite far off in the future, is actually moving incredibly fast, and we were very, very successful in getting very, very large companies to participate. We started the Neural Engineering Industry Group, And since we began the workshops and started this work and got the whole community together, a few hundred people participating across a couple hundred institutions, you've seen Neuralink and Elon Musk's investment of where he's talking about targeting $700 million of investment in this field. Kernel, founded by Brian Johnson, with $100 million. The Mental Typewriter by Facebook. Galvani Bioelectrics is the joint venture with $700 million from Google and GlaxoSmithKline. We have been successful in catalyzing what we see as a new industry. So, if NESD is successful, we expect that within 3 years, we should have a digital interface with APIs to your primary senses. And I would hesitate to say we're going to cure blindness and deafness and aphasia, but we should make a good amount of progress. Cochlear implants are the state of the art today with about 6 to 15 contacts. We're talking about having tens of thousands to millions. Think about the improvement in precision. If we're successful in developing the codes for the visual system, we'll go from phosphenes and blurs of white light to starting to write codes to specific neural encoding elements so that you have sharp edges and fine points and color and motion. And then, of course, for the people that are locked in with ALS and other diseases that inhibit your ability to communicate verbally, we believe we will also be able to read your speech. So what's next? What's next? Well, this is the part where I want to get you thinking and tell you a little bit about what we're working on at Cortical. Our goal, in a sense, is beyond restoration. When I was at DARPA, the challenge was, how can we heal the infirmities of the damaged mind? Loss of eye, loss of ear, stroke, neurological deficit, Parkinson's. But I think thinking beyond that, the next generation, so for you technologists who are developing the next generation of processors and switches and memories and neural networks and learning systems, our goal is to free the mind from the limitations of even healthy bodies. What might come beyond restorative prosthetics, whether it's digital telepathy, being able to read out what someone is intending to communicate or communicating directly with them electronically through one of these interfaces. I talk about virtual reality, where you could begin writing information displayed directly into your senses without any other machinery than your implant. That's coming. And there have already been experiments in animals of collective cognition, of monkeys with their prefrontal cortices wired together so that together they can do a task better than any 3 individual monkeys. It's coming for us too. And when we talk more specifically about what are the next generations of artificial intelligence architectures going to look like, if we take what we learned in making these artificial systems to connect to the brain, what do we know? Well, there's a couple of core areas that I can point to that would be transformative if we can enhance them. And these are the areas that Cortical is focusing on. I think that this paper by Baum and company had a great title. Modular segregation of structural brain networks supports development of executive function. So as human brains and of course other primate brains mature, they start out deeply and densely connected all to each other, kind of like most of our deep learning networks are today. But as you grow older, you go through puberty, your teens begin to develop executive function. It happens through functional decomposition and abstraction into different unique parts of the brain that accomplish and represent different things. And it's through that isolation and through that balkanization into more complex architectures that the more complex brain capabilities emerge. It is literally true that your preteens, so your tween children, you can yell at them and say, "Don't you ever think ahead? Can't you imagine the consequences of your actions?" And the answer is no, they don't have the physical machinery to do those computations. They literally do not have it. So you can rail about it as a parent all you want. What you really have to do is just be a good example and wait until the circuitry emerges. So what I would say is that our deep learning networks today are kind of prepubescent components of a thinking system that haven't gone through the maturing process yet. But let me be a little more specific. One of the key areas that Demis Hassabis and the DeepMind people are starting to poke around in is what I would call assembling sub-AIs into larger integrated meta-AIs. And so when you start to look at the individual systems like DeepMind or Watson that are these knowledge stores, they're very bespoke. It's almost as if these systems are taking some analogies to some tiny little subsystem of the brain, and they're developing that functionality. You look at Alexa and Siri and the Google voice translator. These are systems that are emulating just a small portion of the auditory cortex. The Facebook and Google image identifiers are using convolutional networks to mimic a tiny portion of the visual cortex. But no one yet has successfully built the center of all those things. And by the way, in case you're wondering where this art came from, this is a wonderful piece of actual art by Greg Dunn that was taken from an actual cross-section of a real brain showing the connectivity between the brain regions along a plane that kind of cuts you almost right down the middle. But that integration and synthesis component, you can see that every single part of the brain is connected to that central area. That area is called the hippocampus of the brain. And we used to think that the hippocampus was kind of some of the machinery that would store memories. And it does to a certain extent, but it turns out that the way your brain really works, as we're starting to piece together, is that it's almost like a switching network that activates the entire machinery of your cortex. And when you're remembering something, or when you're trying to process or recognizing something, it's actually the activity of your entire brain, different parts of it lighting up and different strengths to characterize different representations. But it's through this integration in the middle that everything translates from one domain to the other. We talk about our AI not being able to generalize out of its domain. How do we synthesize auditory and visual information? If one of you was to speak in the audience, I probably couldn't hear you, but if I look very closely and I can barely hear you and I can see your lips moving, I will be able to integrate the visual information of what your lips are doing with the bare amounts that I hear and estimate much better what you're saying. The same is true for every context relative to our memories and how we compare what we've learned with what we're seeing. All of this is an integration that assumes one very important thing, which we haven't done in our learning systems yet. It requires that there's a common code that is semantically relevant to both sensory and memory integration. But our deep learning systems don't really do much of that. So I think this is one of the areas where we're talking about building a common code that has general representation across all the senses and memory systems so we can begin to compute meaningful things with learning systems there. So building an artificial hippocampus is the first challenge. The second one that I want to talk about is anticipation. And here, this little fist in the bottom, this is your cerebellum in the back of your brain. It's about a fist-sized chunk of tissue. Now, when I was learning neuroscience, as an undergraduate and graduate in the 80s, everyone was pretty convinced that your hippocampus was involved in motor control. And in fact, you talk about muscle memory, learning to ride a bicycle without thinking about it, skating without thinking about it. Trajectory estimation, the ball is coming at you, you need to catch it. And it turns out that that's true. And if you start to look at the kind of integrated components of the hippocampus. I'm going to come back to some of these. And the cerebellum. Let me go back to that. This is what the cerebellum looks like. It's these incredible sheets of neurons all in a line. And it's a system which learns trajectories. So the neural signals flow perpendicular to these sheets. And as you learn a new routine, a new dance, a new language, how to speak, how to talk, how to move, the connections between the pattern generators and these sheets are refined. But the mind-blowing aspect of the cerebellum, the thing that we've learned just in the last couple of years, is that fist of neurons in the back of your head actually has more neurons than the entire rest of your brain. it just doesn't make sense. It didn't make sense that all it was doing was motor control refinement. And it turns out that was not, in fact, all it was doing. Because the cerebellum, it turns out, is not just connected to your motor cortex, it's connected to your entire brain. So we now believe, or I should say some people, and I myself believe, that the core function, the unitary function of the cerebellum is actually quite simple. It's to project future state. But not just of your muscles and your body, it's to project the entire state of your cognitive processes. What you're feeling, where you are, who you're looking at it, what word do you expect to hear, what is the next thing that's going to come out of my, you all know what I'm going to say. And I'm surprised. And now I'm paying attention because you guessed something different. But that mechanism to project your future state, to anticipate what is going to be, it turns out is fundamental. And so we look at this sort of opportunity to say, what if we could build an artificial cerebellum? And it turns out that for the neural interface work, it was critical to make the thing work at all. And here we were just looking at a kind of a motion capture labeled monkey as he's walking on a treadmill. We had his muscles wired up. That's the red signal in the top right. You had the neural state bottom left and the brain implant to look at the code that was generating it. And so we were able to translate between all these domains. But the performance of the artificial system sucked as long as we looked at fixed state and synchronous snapshots of the state of the brain. It wasn't until we looked at the trajectory of systems that we finally understood the role of time, the importance of anticipation, the projection of the future state. So our goal at Cortical is to extend this trajectory estimation to the entire cognitive arena in all the type of mental processes you do, and we expect significant enhancement across all of them. So let me start to close with a couple of provocative questions. It's not really a question of if this is going to happen anymore, because we're already building the machinery to do it. Within 3 years, we expect some of this to be working. What do you think is going to happen to written and spoken language when instead of just looking at the phonemes at the auditory cortex level, we can begin to look higher in your object representation level in your brain? What does telecom and media look like when we can have digital APIs not just to audio and video, but to your actual senses and emotions? What does your relationship with your spouse look like? So, our goal is to be able to solve problems we couldn't solve by integrating the senses, by embodying fundamental cognitive abstractions in how the patterns of the brain work, and to be able to anticipate and imagine future consequences. But that simple set of 3 advancements, even if they're not superbly functional in their first iteration, is kind of the rudiments of having value judgment and ethics. So the machinery that we need are the processors, the graph representation, the systems to emulate and understand and implement temporal codes to be able to represent these abstractions. Now, once we can emulate one of those abstractions, we can start to spawn recursive versions of it. So one AI instance can begin to spawn other AI instances and anticipate how your own actions and decisions influence others. And I would argue that this is the root of digital empathy. And fundamentally, in order to be able to transcend the current AI systems, which today are more or less tools that we use, and embody them with sufficient trust that they can anticipate the consequences of their actions, they can recognize what's good and bad for them and others, we won't be able to move into an actor that we trust. But here I'm going to leave you with my last provocative suggestion, that when you think of yourself as a conscious being, what is the root of that consciousness? And I've been working on this for the last year, that I think that the fundamental mechanism that underlies all the complexity is the future prediction of your conscious state. The machine is not magical. It's something that's just continually estimating what you're about to feel, what you're about to see, what you're about to hear. And if you take this action, what do you expect to happen? That is consciousness. I don't think it's any more complicated. So I think, and I'm being very careful here, I'm not suggesting that we're going to go out and make a conscious machine. But if we can make an artificial machine that can begin to appreciate some aspects and embody some aspects of ethics and consequence and earn trust, that's when I think AI is really going to be powerful. Thank you very much.
All right. So we've gone a couple of minutes over, but we were having so much fun back that we decided to let Phillip go a little longer and to give him an extra 5 minutes for questions here. So if you do have questions, please get to the mics. We'll be about 10 minutes behind, and we'll squeeze it out of one of the breaks. Thanks.
Anything, anyone. Ask me anything. Yeah.
Charlie Demergent, Semi-Accurate.
Yeah.
Have you or do you have any plans to test any of these implants on yourself? And if not, why not?
That's a great question. Rev 1, probably not. There's a natural dynamic of risk-reward depending on what sort of a deficit you have. The early implants, require invasive surgery, and the FDA wouldn't even let that happen to someone unless there was a tremendous benefit to be earned for the risk. So the early people are the paralyzed, the paraplegic, the blind, and so on. But we do fully expect that every year they'll become more integrated, less invasive, and eventually non-invasive. And at that point, there's no FDA involved anymore, and you can imagine, in maybe 3 or 4, maybe 5 generations of having a general brain implant that's successful without a regulatory process.
Okay.
Shai Galon, Cavium. If you define consciousness as the ability to predict, do you think that computer programs like viruses that predict the next, like red code, for example, are conscious?
I would say in a rudimentary way, yes. The question is, what's the level of complexity in what you can predict? And that depends on how differentiated and how complex the prediction machinery is, what abstractions it can represent and move forward in time. So I would say very limited predictive power in the virus.
But if that is conscious and we're talking about empathy, digital empathy, morality, does that mean that killing a computer virus is actually a crime?
I suppose if you're a virus bigot, that probably wouldn't be judged badly at this time. Anything else? Yeah.
Hi. I was thinking about, if you can figure out the electrical impulses behind a thought, and if you can feed the same impulses to some other brain, would the process remain the same?
Well, the architecture is under your control. And, I think that one of the challenges in developing these neural transcoding systems is really understanding the role of randomness. And I think that a lot of neuroscience has confused what's really happening randomly in biological systems and the probabilistic systems they're using to describe them. So in the former, there's a naturalistic function which has a very repeatable behavior that's not random at all. The latter is a statistical description because you've got limited sampling capability. So there's an inclination to use the sampling technique to try and make something deterministic, but that's an inverse mapping that doesn't really work very well. The randomness that's really happening in biological systems is more related to how is the individual biological computing element synchronized with the external stimulus that's happening at any time. And so it might have been active or been quiescent and recharging. So there is randomness in what you expect to happen depending on time synchronization. But the machine is very, very repeatable, especially in its aggregate shared computing function. So deeper conversation, but find me after and we can talk more about it. Thank you. Last, maybe 2 more questions? Yeah.
Hello, Dennis Nguyen, Cisco. So the ability to make a neuron fluoresce upon stimulation and to make to activate a neuron by illuminating it. That sounds like a big breakthrough all by itself. So what is that? Is that a plasmid? Is that CRISPR? Or could you give more detail about that?
Yeah, so essentially you're talking about a DNA instruction that synthesizes a protein using ribosomes that are part of the cellular machinery. How you deliver that DNA instruction, there's a variety of techniques. You can use viral injection, so where you take a virus and you empty out its natural virus instruction and you cram the payload with your code, and you give the virus, and you have to design the virus so that it heads to the tissue that you want, or maybe control the injection locally. There are other technologies that use mRNA instead of DNA, so it's a temporary thing that can be amplified and control in duration. And there are some where you just spray it on and it diffuses in, although that's a little bit more challenging in certain parts of the body. But again, longer conversation. There's probably a dozen methods, and they're growing by the month.
All right. Unfortunately, my cerebellum anticipated that the talk is over, and we're out of time. So can we thank Dr. Alvelda one more time?
Thanks, everyone.